appears that we are officially live. Hello, everybody, and welcome to another session of our Sussex Vision seminar series, as always within the Worldwide Neuro Initiative. I'm George Kafetsis, a master's graduate from Thomas Euler's lab, and currently a PhD student with Tom Baden. As your host for today, I would like to once again begin by thanking Tim Vogels and Panos Bozelos for putting forward this ever-expanding initiative towards a greener and much more accessible seminar world, without which meeting someone in Hawaii at 7 a.m. would not have been possible. Having said that, allow me, of course, to get back to the reason we all gathered here for today and introduce our guest from University of Hawaii, Professor Megan Porter. Megan's interest in aquatic ecology and phylogenetics appeared early in her academic career. Following her bachelor and master's degrees from Wittenberg and Cincinnati universities, respectively, Megan joined the Brigham Young University, worked with Keith Crandall on crustacean phylogenetic systematics and Opsin evolution, and obtained her PhD in 2005. The same year, she moved to University of Maryland and the lab of Thomas Cronin, where she delved deeper into the Opsin evolution of the complex visual system of stomatopod crustaceans. In 2012, she joined the University of South Dakota before moving to University of Hawaii in 2015, where she has been located ever since, nowadays holding the title of Associate Professor at the School of Life Sciences. Today's focus will be a quite complex visual system, but in her lab, they additionally investigate the evolution both of simple light detection and of eye loss. I could go on and describe the tools they exploit in their holistic approach, but the title of her talk hints at them nicely. Therefore, today we have the opportunity to hear about their latest and I'm sure exciting findings in her talk entitled The Evolution and Development of Visual Complexity, Insights from Stomatopod Visual Anatomy, Physiology, Behavior, and Molecules. So without any further ado from my side, please all welcome Professor Porter. Megan, the stage is officially all yours.
Thank you. Let me just pull up my talk. Okay. Thank you, everyone. Good morning, good afternoon, good evening, depending on where everyone is at the moment. This is really exciting to be able to talk to an international crowd about my research and some of the work that I've been doing more recently. And so let's get into it. First, I wanted to start off with some thanks. None of the work I'm going to present to you today would be possible without a team, an army of fabulous people and collaborators to work with, but specifically the mantis shrimp work, the stomatopod work that I'm going to tell you about, thanks to Justin, Tom, and Roy, the triumvirate of stomatopod visual ecology, where I learned first visual ecology, special thanks to Tom, and also everything I know about stomatopods. Thanks also to Mike and Kate, who were all in the lab, in Tom Cronin's lab together, learning and studying stomatopods. And we continue to collaborate today on trying to understand these crazy visual systems. I wanted to start off by, excuse me, also thanking Worldwide Neuro for the invitation to tell you about my work. You know, there's a great series of talks out there now, including talks by two of the people that I have collaborated with, Justin Marshall and Mike Bach. So you should go check out their talks if you haven't seen them. And they set the stage for what I'm going to tell you. So Justin talked in his Worldwide Neuro seminar a lot about the recent investigations of the neuroanatomy of stomatopod visual systems. Mike talked in his some about color signaling. And what I'm going to tell you about today is connects those two. So a lot of my work focuses on the opsin protein. So opsins are proteins. They form the visual pigments, sort of they're covalently attached to a chromophore. Together, those form a visual pigment, and those visual pigments are responsible for light detection in all animal visual systems, as well as light detection in other contexts as well. But in mantis shrimp or stomatopods, the color signaling that Mike talked about It's these visual pigments, these opsin proteins that are responsible for detecting those different wavelengths of color and then initiating the cellular cascade, sending the signal down through the neural processing centers that then provides the animal with information for appropriate behaviors. So there's a link there in some of the seminars. To tell you a little bit about my lab, the Porter Vision Lab, we study vision in a whole range of animals, including a lot of marine crustaceans, which is where my heart lies. But we have been working on things like worms with Mike, which he talked about in his seminar. We work on a number now of terrestrial arthropod systems as well. And every once in a while, a vertebrate sneaks in there. There's a student in the lab now, a PhD student studying seabird vision. So diversity of different animal systems, all trying to understand sort of how the molecules of vision fit into a greater understanding of how the visual system works and what an animal is perceiving, how they're using that information. And in thinking about this talk, thinking about my work and how to present it, I realized that there's sort of this theme as well in a lot of the work that we're doing in the lab. And that's getting at this idea that in many animal visual systems now, it's been documented that there is a mismatch between what we know about photoreceptor diversity and what we are learning about expressed opsin diversity. And some examples of that. So here are just two examples of that for more recent literature. On the left, from dragonflies, from a paper in 2015, that's a phylogeny of opsin diversity across a number of species, broken down by what type of visual pigments those opsins form, whether they're UV sensitive, short wavelength sensitive, so in the blue parts of the spectrum, or long wavelength sensitive in the green parts of the spectrum. And you see a range of just opsins thought to be involved in vision, anywhere from 11 up to 29 copies expressed in these eyes. That's a lot of opsins for what we understand about how arthropod anatomy, visual anatomy, the diversity of photoreceptors in some of these systems. So there's a big mismatch there. It also happens in vertebrates. So a recent paper as well in the deep sea fish in the silver spiny fin. They found 40 opsins in the genome, which is a crazy number of opsins per vertebrate. And it showed that at least 14 of those are expressed. So across a large number of animal systems now, thanks a lot to the, as tools, the genetic tools for investigating opsin expression have gotten easier to use, and we learn more, we can get more information. We see this really big mismatch in the number of opsins expressed and what we understand currently anyway about the types of photoreceptors in these animal visual systems. And so that suggests to me two things. One, opsins are doing some things that we don't understand yet in visual systems, but also maybe there's some hidden diversity in photoreceptors that we haven't uncovered yet based, using the physiological and anatomical studies that these have been characterized in the past. So what I'm going to tell you about today are my studies trying to reconcile this mismatch in crustaceans. And I'm going to tell you mostly about my work in stomatopods, also called mantis shrimp. You'll hear me go back and forth with that between the scientific name and the common name. But also I'm going to tell you a little bit about sort of my new crustacean obsession, which is copepods. Copepods, I think, are another fantastic group for studying the evolution of visual system diversity. I'm going to just tell you a little bit about that and hopefully convince you that everyone should love copepods as much as I do and should consider them for their studies. And before I get into the details, I just want to thank all the people in my lab who, in addition to the people I've already thanked, the people in my lab, a lot of the recent work is due to their spectacular research and their questions and just their interest and passion for science. And so thanks to all of them. In particular, I'm going to show you some of Sitara and Marissa's dissertation work towards the end of the talk. Okay, so to start off, I want to take a broad stroke overall view. As I said, I work mostly in crustaceans, and when I say crustaceans, I mean the pancrustaceans minus the insects. Taxonomy has changed a little bit, but I want to take a broad stroke view, both of the sort of diversity on the anatomical side, the morphological side, and the diversity on the molecular side, and then use that as a place to start drilling down deeper to try to figure out these mismatches that we're seeing. So on the morphological side, this phylogeny is one that was published a number of years ago now, and it just shows across crustacean lineages the variation in the design of the compound eyes. And I'm not going to go into the details of that other than to say that the differences that you see here are mostly due to the optical structures that are focusing light onto the underlying photoreceptors. But this simple sort of broad stroke view misses a lot of the complexity and the variation in the visual systems and the way that those are put together. And you can see that just in the sort of small selection of crustacean eyes I have there on the right. And as an example of some of the hidden diversity when you take a broad stroke view that you find when you start to drill down deeper into some of these lineages, I'm going to talk a little bit about the copepods. So copepods are typically one of the crustacean lineages that are thought to have a simple eye. And that simple eye has been referred to in the literature as a Doppler eye or a tripartite eye because it has three receptor cups. Here is a cross section of it, sort of frontal cross section. Here's a lateral cross section. It's got paired sets of ocelli or photoreceptor cups that are dorsal, a single one that is ventral, that are sort of all merged together into a structure. But if you look at the eye morphology across different species of copepods, and this is just one order of copepods, an example of some of the morphological diversity in eye structures of that simple eye. The structures are colored the same. So the paired ocelli, dorsal ocelli, are in sort of salmon color, the unpaired and sort of this dark red. There are lenses in blue. You get a sense of the modifications to the simple structure. So some species have greatly enlarged the dorsal set of receptor cups. Some have greatly enlarged down here the ventral set, single pigment cup. Some have pulled apart those three cups into separate eyes. Some have added lenses or multiple lenses. There's sort of a dizzying array of eyes based on just this simple design in copepods that I think is really interesting evolutionarily and needs some further work. And we're starting to dig into how the molecular components compare with this as well. But they're a fabulous system for studying the evolution of visual system diversity. Okay, and that's just as an example of all of this diversity, and I think diversity in the anatomy of visual systems and the underlying photoreceptors that there's still a lot that we don't understand. On the molecular side, which is where most of my work has been, we are also wanted to start with a broad stroke brush and just look and see across crustaceans, again here across orders, what types of opsins are different groups of crustaceans expressing in their visual systems. And to do this, we took published data, transcriptome data, and just searched through for expressed opsins. And for each species, identified the non-visual and visual opsins as we understand them based on evolutionary placement, to get a sense of what lineages are expressing what opsins. And it looks like overall, crustaceans have, if you look just here in this part that's the visual opsins, They seem to be expressing long wavelength options of a clade of long wavelength options. They have 3 clades of what we typically think of as middle wavelength or blue sensitive options, noting that the barnacles sort of have their own clade. And then a clade of, in purple here, indicates short wavelength, probably ultraviolet options. So again, this is a broad stroke view, but when you begin to drill down, most of the species that we're looking at least many of them, you look at the numbers of options expressed in each of these groups. There are many more than we would predict based on the little bit that we know about the photoreceptor array in their eyes. So what's going on here? And I think the work that I'm going to show you is attempting to drill down from sort of both sides, both morphology and molecular, adding in some physiology as well, to try to figure out where that mismatch is and use those mismatches to identify sort of hidden visual system diversity that we haven't yet characterized. Okay, so with that as the broad stroke foundation overview of some of this work, let's get into the main part of the talk, which is the evolution and development of visual complexity in mantis shrimp. And I like to start off with this picture by Roy Caldwell, because it really, I think, clearly demonstrates the differences between larval and adult eyes in this group. So here you have these giant eyes of an adult lysosquilloid species, which lives in a sand burrow, so it's just poking out of its hole. And in between that, you have an entire larval individual with its tiny eyes. So there are big differences just in size, but the eyes are different in a lot of other ways as well. Stomatopods, I think, are an interesting system for looking at the development of visual systems for a lot of reasons. One, we can get this to go. There we go. We can collect clutches of eggs. We can watch those eyes develop, so you can catch different stages of eye development. We can hatch those out into early stage larvae, which we then can study using various molecular, anatomical, physiological techniques, as you'll see. But they're also interesting developmentally because the larvae, up until recently, were typically thought to have a visual system like any other crustacean larvae, which is of a type called a transparent apposition eye, where the optics are sort of stretched out and the pigmented receptors are condensed to as small an area as possible, without any diversity in those photoreceptors. So we tend to call them simple eyes. So if we put that all together into the life history of a stomatopod, we have these complex adult eyes I'm going to tell you about in a minute, where I've spent a lot of time trying to understand how the molecules fit into what we know about the visual systems. We can catch the development of that visual system into this simple, a larvae that has a simpler eye than the adult anyway. And then we have this transition between this simple eye and this adult eye, which is another feature of stomatopods that makes them the visual systems interesting. And that's that the larval eyes are physiologically and morphologically distinct from the adults. So here's a close-up of a pair of larval eyes. And this particular larva is in the process of transitioning from a pelagic larval form, so something that lives up in the water column. It's metamorphosing into a juvenile and transitioning to the benthos. So adults live on the bottom of the ocean. And what you see happening here is that they don't transition directly from the larval eye to the adult eye. They just build an entirely new adult eye right next to that larval eye as they're making that transition. So you see here this sort of blob in pale yellow is the larval eye, and on top of that, it is building a completely separate and different adult retina. So it makes this really interesting system because we have very distinct eyes. the larvae and the adult are in very different habitats and ecologies. And not only do they build new retinas, here is a schematic that Kate Feller put together. Along with that adult retina, it's also building new neural processing centers, at least through the lamina and medulla here in orange, whereas the larval is here in sort of pale yellow. So there's some questions here about how much of this neural circuitry is recreated for that adult retina, when and where does it get wired into the existing circuitry? Those are questions we don't know yet, but it is part of this complexity. It's a piece of the puzzle that we're, hopefully one day we'll understand. Okay, so that sets the stage in terms of the life cycle and the visual system between larval and adult mantis shrimp. So I wanted to start off with the complex adult eye, because that is where most of what we understand about mantis shrimp visual systems comes from studies of adult visual systems. So if you've never seen a mantis shrimp up close, this is what one looks like, and an even closer view of its eye. They are crustaceans, so they have compound eyes, which means they have individual units, but they're very different from other crustaceans and other arthropods in that you can see here from these dark patches, which are pseudopupils. The pseudopupils are the parts of the eye that are looking directly at the observer. So you can see that there are 3 parts in this eye looking directly at you, which means that they have trinocular vision. They have depth perception with one eye. And the reason is because they have this specialized set of ommatidia that run around the equator of the eye that we call the midband. And in the most complex eye forms, there are 6 rows of ommatidia in that midband. So what do the underlying photoreceptors look like? If this is the sort of most complex eye in terms of external morphology. And to understand that, first let me tell you what a typical crustacean eye looks like. And then we'll get to the adult. So a typical crustacean eye, it's compound, it has those facets. Each one of those facets represents a single optical unit we call an ommatidium. If you take a cross section of one of those, there are some optical structures on top, light coming in from the top through a cornea, a crystalline cone, which focus light onto receptor cells underneath. And crustaceans typically have 8 receptor cells. A single cell that sits on top we call the R8, typically sensitive to violet or ultraviolet light. And then the remaining 7 cells work together to form what we call a main rhabdome, a photoreceptor, where those 7 cells form a tube and project microvilli into that tube. And that's where the visual pigments are found. So light travels down through these photoreceptor cells, and that's where you get the light detection. So if we go back to stomatopods and we look both anatomically and physiologically at the receptors underlying that sort of overall eye design that I talked about with the midband rows. So now we're looking at this cross section. Here are the 6 midband rows. Light is coming in from this direction. We have this ommatidium here represents the dorsal part of the eye, the dorsal hemisphere. This represents the ventral part of the eye, or the ventral hemisphere. Each of those photoreceptors is colored based on the measured spectral sensitivity. And there are at least 16 different types of spectrally different photoreceptor. I think Justin Marshall is arguing for a few more these days. And I want to break down how we get to some of these differences. But I also want to point out, I've been using these terms a little bit already, but I'm going to be saying ultraviolet sensitive to refer to sensitivity or absorbance on this part of the visible spectrum. Middle wavelength sensitivity sort of in the blue range, 400 to 500 nanometers, and long wavelength sensitivity sort of to anything above 500 nanometers. All right, so if we break down that diversity, stomatopods have, in those R8 cells, in the most complex size, 6 different types of ultraviolet photoreceptors. Interestingly, from Mike Bach's work, those photoreceptors are due to 2 types, that's 6 different spectral sensitivities or photoreceptors are due to sort of absorbance of 2 types of ultraviolet RH photoreceptor cells and 4 different ultraviolet filters found in the crystalline cones. So 2 photoreceptors, 4 filters, put together in different ways equals 6 types of photoreceptors. The main rhabdomes, those R1 through 7 cells in the hemispheres of the eye, are thought to be used for spatial and motion vision, and based on this, they have a single spectral sensitivity in the blue range of the spectrum. In rows 5 and 6, those main receptors, there are 2 receptor types. They have the same spectral sensitivity, but differ in anatomy. So the microvilli are oriented differently in those 2 sets of receptors in rows 5 and 6. And then in rows 1 through 4 of the midband, those 7 cells that I talked about have been subdivided into 2 sets of photoreceptors, each with a unique spectral sensitivity. And together, those 8 receptors are what are used for color vision in mantis shrimp. And I should point out that the spectral tuning of those photoreceptors is also due to some filtering. So in the most complex visual systems, there are also 4 colored filters that sit between photoreceptors. All right, so that's a lot of pieces to put together. And that's just for the most complex visual system in stomatopods. But you know, we know if we look across species that there are variations in all of these components. So here's just 3 species. You can see variations in the size and shape of the eye. There are also variations externally in the number of midband rows, which says immediately that there's variations in, you know, the complexity of the photoreceptors underlying those visual systems. So one of the things that I want to do to try to set the stage for the morphological anatomical complexity is to put this all together in an evolutionary sense. So taking a phylogeny from some of my earlier work, there's 7 families represented here by the sort of single letter codes. I'm going to put together all that information. So first of all, just the number of midband rows varies across species, and in this particular set, anywhere from 2 to 6 midband rows. Those seem to be the most common types found in stomatopod visual systems. Underlying that, we have the number of photoreceptor types that I just walked you through, anywhere from 1 to 16. But contributing to that, we already know that there's differences in the number of visible filters between those photoreceptors that play a role in those photoreceptor types. So there's variation there. There's also variation in the number of filters on the UV part of the spectrum from Mike's work. So to me, when you put this all together, first of all, looking at just the variation in the number of ultraviolet and sort of visible light filters, it suggests to me that maybe we need to go back and revisit our estimate of photoreceptor types. There's some variation there, I think, that we haven't put together yet. But for the most part, all of these pieces make sense based on what we know about the physiology of visual systems in mantis shrimp. And so this is where I wanted to start plugging in the molecular data to see how that fits in. And over some number of years now, I've been collecting transcriptome data from a number of species to try to get at this. And so when we plug in the number of expressed visual opsins, it stops making sense again. So we have, in many cases, many more opsins than predicted based on what we understand, again, about that photoreceptor diversity. Sometimes twice as much. Here, for some of the species expressing 30-some opsins. But also they are varying in sort of unpredictable ways, so which means that there's something that we don't understand yet. Given these numbers are squishy, right? So they're estimates, they're from transcriptome data. The assembly process is difficult, but I think they are hinting at things that that we don't understand about this mismatch between photoreceptor and opsin diversity. But you might say, wait, Megan, there are some species that look like they match up pretty closely. But if you pull that apart and look at the number of opsins expressed in each spectral class, whether that's ultraviolet, middle wavelength, or long wavelength sensitive, there's still a mismatch between number of opsins expressed and the number of photoreceptors that we think is there based on the information we have so far. So what's the next step? That really is to go into one of these species. We chose Neogonodactylus oersteri, and to figure out where all of these opsins are expressed in the eye to give us the next piece of information about this mismatch. So just to remind you, this species, Neogonodactylus oersteri, we think has at least 16 types of photoreceptors. It's got 33 expressed opsins. And the next piece of information I'm going to show you is in situ hybridization data for the expression patterns of each one of those opsins across this sort of retinal schematic. So we'll start off with the ultraviolet sensitive opsins. There are 3 of those, and this is again from some of Mike Bach's work. In the expression of these opsins, there were 3. 2 of those opsins lined up really well with Mike's work from measuring photoreceptor absorbances. They fit that pattern, but we still have this one that's expressed that doesn't. We weren't able to find it expressed in the eye, so it's unknown what it is doing, whether it's expressed someplace else, maybe in a neuroprocessing center. So there's a mystery there that we're not quite sure what that particular opsin is doing. If we jump next to the expression patterns and the long wavelength sensitive opsins, there were 20 of these in the Neogonodactylus oersteri. I know this is a big figure. The clades, the opsin transcripts are numbered and colored by clade now, rather than anything related to spectral sensitivity. And here you see the actual in situ data, and the schematic is colored to give you sort of a reference point, something hopefully you're a little familiar with, to where that expression is. The takeaway from this really is that most of these opsins are expressed in what we have been characterized as long wavelength sensitive photoreceptive cells. So that is good. That includes things down here like the main receptors of the hemispheres, or in the mid-band receptor cells that have been physiologically characterized as long wavelength sensitive. The thing here is just that there are so many of these opsins that most of these photoreceptor cells are expressing many copies of them. So there must be some co-expression going on, and that's sort of the next step in understanding this mismatch. We also uncovered a transcript that doesn't seem to have a visual function, at least based on this expression pattern. So this is a transcript that is expressed in all 8 of the receptor cells that are responsible for color vision in mantis shrimp. So it can't possibly have at least the idea here is that it can't have a visual function, because each of those receptor cells have a distinct spectral sensitivity. So what this opsin is doing, whether it's turned into a protein, we don't know. And we're actually working on that now, trying to do some protein localization studies to try to give us a clue as to what's going on. And then finally, if we look at the 10 opsins that are middle wavelength sensitive, again, same setup. Each opsin is numbered, colored by evolutionary clade, with the in situ data here, and then the schematic showing you what that labeling pattern looks like. Some of these opsins are expressed in middle wavelength photoreceptors, which is good, was what was expected. But some of them, the remaining actually, are all expressed in what had been previously characterized as long wavelength sensitive photoreceptors. So all of the long wavelength sensitive photoreceptors express at least one middle wavelength sensitive opsin. So there's something going on there in the stomatopod visual system. Again, we need to do some protein localization studies to try to figure out what's going on. We were particularly interested in, there are 3 opsin transcripts that have a very distinct expression pattern just in the very dorsal part of the eye, which is intriguing, particularly given some recent work by Ricky Patel, behavioral work showing that mantis shrimp can use celestial cues to navigate. So we think we have identified using this molecular approach some receptors in the dorsal part of the eye that hadn't yet been characterized. And if you sort of squint at that in situ data, you can maybe convince yourself that the receptors in that part of the eye change direction and sort of point up. So this is a particular part of the eye we're focusing on now that we've identified using molecular expression. Something unusual is going on here, perhaps related to navigation. And so we're working more on, a whole group of people is thinking about that now and trying to characterize that part of the eye. So just to sort of summarize all of that work and where we think we are, this graph, so we have dorsal hemisphere photoreceptors, the photoreceptors of the mid-band, rows 1 through 4, 5, and 6, and then the ventral hemisphere receptors. And then the bars on the y-axis here, just the number of Opsin transcripts colored by clade, as you saw in the previous figures. And this is just to give you a broad stroke idea of the pattern, some of the patterns we're seeing. So if you look just at the receptors used for color vision, they seem to express 1, maybe 2 transcripts, plus this transcript that is expressed in all of them. So they all have sort of a unique transcript plus this common opsin, but they express many fewer transcripts than the rest of the receptors in the hemispheres, which have previously been characterized as long wavelength sensitive. They all express, 6 or more Opsin transcripts of multiple spectral classes, at least as determined by their evolutionary placement. And in particular, we're really interested in this sort of very dorsal part of the eye that's expressing multiple middle wavelength sensitive opsins and maybe related to navigation. Okay, so we've learned a lot about from coming in and adding the molecular perspective to what was known about the way the stomatopod visual system works in the adults. What about in the simple larval eye? Clearly there's still a lot to be done there, but at some point I thought, okay, maybe we should take a step back. Let's look at a simpler system. Let's try to understand what's going on in the larvae so we can understand how we get developmentally from this simple eye to this really complex system. There's been a little bit of work done in these sort of simple larval eyes from a paper from Kate Feller and Tom Cronin. However, I'm characterizing the peak absorbance of different photoreceptors and different the larvae of different species. Most of them seem to have photoreceptors that have peak absorbance in the blue part of the spectrum. But if you look at some of these, there's a huge range in the data points, right? And there's measured values of peak absorbance with some outliers, sort of up really long wavelength, some that are down really short wavelength. So this suggests that these eyes are not as simple as maybe we assumed that they were. And so again, we went in, first pass in my lab is generally molecular. So we went and started to look at stomatopod larval visual systems using molecular tools. And this is some of Sitara Palakanda's work, her dissertation work. And so she started looking at the expression of opsins across development in various species of mantis shrimp. So what you see here are the different developmental stages she looked at in a Poiascula thomasini. A couple of different embryonic stages, an early larval stage. The E here is that transition between larval and adult. So that's a double retina stage, and then the adult. And then below that is the number of Opsin transcripts broken down again by spectral class, ultraviolet, middle wavelength, long wavelength sensitive. So there are some interesting patterns to notice here. First of all, as the eyes develop, they start to express more opsins. That's maybe not too surprising. By the time Poioscola thomasini is a larvae, it is expressing, is that 13 opsins total, including 2 ultraviolet. And that, again, is certainly more than we would have predicted based on what we understood about the anatomy of the eye prior to this study. And then you see, not too surprising, as it's building that adult eye in that transitional stage, you get a jump that is expressing almost all of the adult complement of opsins. And then when you get to the adult, it has about double the number of opsins of middle wavelength and long wavelength as the larval phase. Because of this, this seemed to be where the interesting transition was in number of opsins expressed. Sitara went and looked at another species, Gonodactylaceus falcatus, and found the same thing. So early larval stage, transitional stage, adult. The early larval stage expresses more opsins than we would have predicted, and it's about half-ish of what we find expressed in the adult. So some interesting things about this is notice that for the ultraviolet sensitive opsins, that stays constant. And those are the same opsins in both larval and adult. And in general, what happens, even though that these eyes are separate, all of the opsins that the adults use are just expressing the same set as in the larval form and just adding to that. So just expressing more. So even though the eyes are separate, it's still using all those same larval opsins to build that retina and build that visual system. But again, what I was hoping would be a much simpler visual system turned to be out, at least from the opsin perspective, more diverse than we had predicted. So they're suggesting something is going on. There's sort of cryptic photoreceptor diversity in the larval eyes that we haven't really characterized yet. If we take a little bit closer look at just sort of pieces of some of these larval transcripts, this is again some of Sitara's work. Here's a piece of a phylogeny of larval opsin transcripts. So these are Oesterdae opsin transcripts from that adult study I just showed you. And then the larval ones, and then to the side, we've got red, yellow, or green dots, which indicate whether which stage that opsin was found expressed in. The interesting thing here is that basically all of the stages express all of these opsins. And we know from Oesterdae, at least we think we know, that those opsins are expressed in the midband row. So that's particularly intriguing to be finding opsins expressed in the larval form, but that doesn't have that midband. That in the adult are found in that specialized set of receptors. So I think looking at the expression patterns of some of these opsins in larvae may give us a clue as to how evolutionarily and developmentally stomatopods were able to get to this complex visual system we see in the adults. And we see the same thing in ultraviolet transcripts. So here in Oesterdae, I showed you this a little bit ago. We find all of those opsin transcripts in basically every larva that we've looked at, including that UV3 transcript that doesn't seem to be expressed in the adult eye. So maybe that's a larval specific opsin making a visual pigment just for larvae. We don't know, but we found all 3 of those opsins in the larvae of Neogonodactylus as well. So maybe that will help us unravel what's going on with that transcript. So we found the molecules. Marissa McDonald, who just defended a week or so ago, has been studying this from anatomical and physiological perspective. So she has done a bunch of TEM work to show that, so this is a TEM of a receptor right at that R8, R1 through 7 boundary. And if I color that, so you can see it clearly, she found the presence of these R8 cells that are the ultraviolet photoreceptor cells in 6 larvae from 6 species across 3 superfamilies. So we think that predominantly stomatopod larvae are going to have UV sensitivity. And then she also went in and did some really tricky physiology using ERG recordings, electroretinograms, and some chromatic adaptations. I'm not really going to describe the details of those because we're getting short on time, but she did this in a couple of species, and just to show you, she found physiological differences in the larvae between species. So in Gonodactylus falcatus, she found a peak in the blue at 460, but using chromatic adaptations with different colored lights, she was able to show they also have a peak in the UV at 340, spectral sensitivity peak, and the sensitivity peak in log wavelengths at 581 nanometers. So this matches with what we're seeing with Opsin expression. They have sensitivity in UV blue and long wavelengths. It doesn't necessarily match up to the numbers of transcripts we're seeing yet, but at least we know that physiologically they've got a diversity of photoreceptors. In a different species, Poioscula thomasini, she found that the dominant peak was in the long wavelengths, actually. And this matches up with some anatomical studies that Kate Feller has previously published. But again, using chromatic adaptations showed that there's a peak in the blue at 464 and a sensitivity peak in the UV as well. So if you put it all together, she's done this in larvae from 3 different species now. They all have at least 3 spectral sensitivity peaks. I think the interesting thing here is that, the peaks in the blue are pretty consistent, but we're seeing variation both in where the spectral placement of the peak and the long wavelengths are, as well as on the UV side. So, the stomatopod larvae have visual systems designed for different visual tasks. We just don't understand what those are yet. All right, so to wrap up, to get back to this idea that many visual systems have this mismatch, what have we learned from these studies of the evolution and development of visual systems in stomatopods? Larvae express fewer opsins than adults, but more than predicted. So we still have this mismatch, suggesting we need to go in and look for this photoreceptor diversity and understand what's going on. Larvae express opsins found in the midband receptors in the adults, so structures they don't have, which may give us a clue as to the evolution of those complex structures. They certainly have UV vision, which we have shown molecularly, anatomically, and physiologically. And that by adding the molecular piece to our overall understanding of vision in mantis shrimp, we've come up with cells, photoreceptor cells, with different functions that have different patterns of opsin expression. Some visual opsins have non-visual functions we think that need some further investigation. And we've been able to uncover some of this hidden photoreceptor diversity by looking at those patterns of expression. So with that, hopefully, well, first I have to thank all of my funders for a lot of that work, and again, all of my collaborators. Hopefully I've inspired you to think about, putting all of these pieces together for understanding visual systems, and maybe piqued your curiosity about copepods a little bit and stomatopods a whole lot. And with that, I'd be happy to take any questions.
Thank you very much, Megan, for this very interesting presentation personally, and I hope it's not just a personal view, but like the bigger part of the audience also agrees with me. I find it astonishing, like how complex, even at the Opsin level, their vision might be. I already posted the Zoom room link in the YouTube chat, and the first question that I personally have is the following. So you mentioned that the larvae also have UV sensitivity, right? And we know, like from other animals, that how they use UV differs from animal to animal. Like for example, for mice, you use UV to find predators on the sky. For zebrafish, you use UV to catch your prey. Do you know if the larvae and the adults would use UV for similar goals, or if that would differ completely?
Well, there's a lot of speculation, particularly for larvae. One of the hypotheses is that it is to help them catch prey, to sort of improve contrast in the aquatic realm. Marissa actually has tried some behavioral feeding trials to try to test that. and it's a difficult thing. It's a difficult thing to get at, particularly for tiny things that are hard to find to begin with. It could be related to, circadian rhythms. There were some ideas that maybe it's related to movement up and down in the water column, for larvae in particular. In adults, I think it's being used in a completely different behavioral context, probably for a lot of Mike's work, and Amanda Franklin's work shows that it's probably being used in aggressive interactions.
Right.
So
in the behaviors.
Yeah, in the behaviors. So I think it's interesting. They're using sort of the same sets of opsins in very different behavioral contexts. So it'll be interesting to see how from the opsin work, how the photoreceptors, those different opsins are expressed across the eye, if that'll give us a clue about how they're using them.
Right. Thank you very much. So the first question appearing in the chat is from Thomas Cronin. Megan, are there no larva-specific opsins that you have found?
No. There's maybe one. There's one possible one, I think, in Gonodactylaceus falcatus, but the majority of the opsins that we're finding in the larval eyes are entirely, that entire set is used in the adult eye, and then they just add some more on top of that.
Yeah, his emphasis was also like because he highlights the no larva-specific opsins. So the next one is from Simon Laughlin. How fast do rhabdomers turn over?
That is a great question. I have no idea. I would love for somebody to study that. we can get you some larvae if you'd like to look at that. Adults as well. I have no idea.
Simon, if you want, you can already join us in the Zoom room. And actually, like his question kind of takes smoothly to one of mine. So isn't there even indirect evidence of potentially different kinetics in photoreceptors, like from transcriptomics or something? Because like we know about opsins quite a lot, like confusingly a lot, but what about the rest of the molecular machinery in these cells? So from some of the earlier work that I did with opsins expressed in adults, some of the sort of studies of selection in these suggest that sites under selection are not related to spectral tuning, so in the chromophore binding pocket, but more related to G protein binding. So my sort of hand-wavy hypotheses based on that is that, yeah, I think some of these transcripts and the number of transcripts that we see may indeed be involved in fine-tuning kinetics of the photoreceptors. But I think I'm a long way off from being able to really get at that and test that in any real way. So yeah, I think there's a large potential that is something that is going on. I'm pretty far away from being able to test it. Mm-hmm. And Thomas Cronin adds as a remark to your answer, most likely, that it's the situation that you are describing is very different from dragonflies, for instance. So yeah, like another question I have is like, because you mentioned that they create a new retina for the adult, and this is only for the retina, it's not like for the lamina modula, or is it all, like altogether is recreated? So when they make that new adult retina, it also appears that they make a new lamina and a new medulla to go with that. I don't how much farther down the neural circuitry. I don't know. Like it becomes a black box about how much more is of that new circuitry is needed to process the complex set of photoreceptors in the adult eye. Right. So it needs entirely new processing centers to be able to handle that information. I think there's a question of how much is built new for that new adult retina, and when and where does that get wired in to the existing neural pathways? We don't know yet. And I guess no one has actually tried like to see if these retinas have the potential to regenerate like after injury, as we see in other scenarios. So I think similar to other crustaceans in stomatopod adults anyway, if they lose an eye, they grow back an antenna. So they're not capable of regenerating the eye. Okay. Very interesting. So at this point, Megan, if you want, you can stop screen sharing, even though the eyes are magnificent, just so we appear on people's screens bigger.
Okay.
And I will post the Zoom room link again. And I would like to thank our audience for being here with us. I would like to thank you, Megan, for this amazing talk. And I will be stopping the live broadcast after my next question. So if anyone wants to keep track or participate in the brief conversation we will have, please make sure you follow the Zoom room link that I just posted. So one last question I have is, like, you mentioned these 7 families, right? So do we know what might be the ancestral kind of form and how similar that is to a larva? Like, do we have the relationship between phylogeny, ontogeny, and so on?
So, I mean, there's a little bit of back and forth on the phylogenetic arrangement of families in stomatopods. The basal lineage, anyway, I think most of us agree on now, it's simpler than the most complex form, but it is not. I don't think we have had the opportunity to study the larvae in that particular group of species. And based on the reconstructions that we've done, I think that the ancestral, at least adult stomatopodi, was probably pretty complex. It probably had 6 mid-band rows. It probably had some subset of those filters. And then the diversity that we see in the group is either building on that or losses of components of that.
Right.
How we haven't done that same sort of reconstruction. We don't have enough information about the larvae across that taxonomic diversity yet. So I think we need to fill in that piece to be able to link those 2 pieces together. We're not there yet.
We need more humans.
Yes, yes, we do. More
be
people
part
need
of
to
these
come
efforts.
and study. We can get you stomatopod larvae. You'd like to study them.
So before I stop the live broadcast, just so because you cannot see the messages, Simon laughing said that, Megan, I regret that I have to leave. Very interesting talk. So much to think about. Thank you. And with that comment from Simon, I will be terminating the broadcast. So thank you once again, Megan, for this fantastic talk. And we are officially offline. So